would always have this done when I went to donate blood until I quit donating blood because I never passed the test. It's called erythrocyte sedimentation rate, and it occurs when there's inflammatory factors such as fibrinity. So most anemias, but it's a test on the rate of settling of the red blood cells, and it's measured in millimeters per hour. And I don't ever remember waiting like a whole hour for this test to be done, but it's something they can do without a lot of equipment. To me, running a hemoglobin is a lot faster. I'm going to do that, hemoglobin or hematocrit, you don't hematocrit in less than 3 minutes, however long it takes to spin the centrifuge. So the red blood cell has negative charges on it, and if there's a lot of fibrin threads that are already formed, we're going to talk about that as part of blood coagulation. So if there's a lot of threads, fibrin threads from those numbers increase with inflammatory conditions, then the red blood cells start stacking together on top of each other, kind of like what we saw with endochondral ossification, and the chondroblasts reproduce too fast and stack on top of each other. So when they stick together like this and stack on top, it makes them more dense and therefore heavier as a group, and they settle to the bottom, okay? So it's just kind of, it doesn't really tell you which of these occurs. It's like, well, doctors do this if you come in with a fever, come in with, it's not going to tell them any more than you have an inflammatory reaction going on, which you can tell them from your symptoms. So, like I said, it's just something I want you to recognize because it's still used, but it just means further testing has to be done, and you were told them when you walked in the doctor's office. It slows down. It's a little thicker, so viscosity with higher viscosity, sickle cell anemia, and congestive heart failure. But again, none of this is very specific. Okay, so I really just want you to be aware of what's going on in the test, but I'm surprised they still do it. What's being tested here? Hematocrit. Okay, so this is that percent of the cell volume over the total volume. We started talking off about that one. And then this is, remember, look at the values. This is in grams per deciliter. Hemoglobin, exactly, yeah. So on a lab quiz or a lab exam, if I were to show you the apparatus and say, what's the blood test that this is being used for, they want to tell me the erythrocyte sedimentation rate or the hemoglobin or the hematocrit. Okay? All right, so quiz number 1, question number 1 on quiz number 15. And during the time that you guys are doing the blood typing before, or during, or after, I'm going to do some oil immersion and practice looking for white blood cells. So got to figure out what that cell is, and then which of those characteristics would you apply to it? It may be one or all or none of those particular characteristics. So I'll increase the size a little bit. I'll have the lab finished by tomorrow night, and I haven't started on the lecture other than running the scantrons for either group, so sometime this week. Okay, I'll have them. Normally I have both all day today and most of tomorrow to grade, but being on the interview committee is taking all that time away. We missed our first interviewee this morning, Bender Bender, and that was it. We don't have any time. Can you imagine starting out your day that way? Yeah. We were even willing to give up our lunch break, but gonna go without lunch, just eat it quickly, you know? And then so I said, well, some people get angry when they don't have their food, so that would be fair to the later candidate. So I didn't really talk about size, you might need to Google that. Talk about numbers, how frequent they are, but I didn't really talk about size. Oh, yeah, okay. I didn't write it on the board, so I wasn't sure.
Oh, I'm pretty sure it does.
Just put a white one. Or what, our packet? There's the lab, and here's my number.
I'm pretty sure it is, sir.
Okay, so we want more time on number one. There's one or four or none? The statement is three.
Or is it a weak side? If it is, then
Okay,
it's
we can come back to this. The question number two is the same question, but with a different cell.
No, that's yeah, it has a piston, but it doesn't break down. So
You want to compare?
Yes.
okay, it's
Your answer choices are the same. Okay, this cell. You want to go to basic cell or stay on this one?
two
So
is
to
8 and 9 is
compare,
9.
you should be able to see a distinction. Okay.
Now we can.
I think 1 is not 8. Let me choose. Most likely 8. That 1 is just 2 is just 8.
They are online. In the original diagram. Okay, ready to move on? So we're moving to page 32, and then we'll go back after lab and talk about what hemostasis. So we're gonna look at blood type, which is based on the presence of, for our scenario, it's gonna get a little more complicated, the presence or absence of 3 antigens. And we don't talk about antigens until next week as far as the definition and so on, but we're talking about antigens and antibodies tonight. So an antigen is a molecule that triggers an immune response. An immune response is the response of the body to an antigen, which is called circular reasoning. Doesn't really tell you anything. But antigens are typically, if they're too small, they're not going to trigger an immune response. All right, so if we're trying to create a vaccine against, you're in science and you're trying to make an antibody so that you can select out certain molecules in a solution, and a molecule is too small, you can attach it to a larger molecule and inject it into a mouse to obtain antibodies, and it will be to all sorts of things, including the larger molecule, but you at least can get antibodies to the molecule you're selecting. So the antigen here, in the case of the red blood cell, is a surface substance. These can, for any antigen, it can be a glycolipid or a glycoprotein or a protein, and it's going to be attached to the cell surface, or it may not be. All right. So by identifying the antigen, that is the step towards identifying the blood type. We identify the blood type for the antigen that's present. So if the A antigen is present, then this would be blood type A. If the D antigen is also present, then we add that name to our blood type, and this is blood type AB. If the D antigen is present, that's also known as the positive or negative. If you're identifying someone's blood type as AB positive or AB negative, that is the D antigen. And so this person would be AB positive, okay? If they only have the B, they would be B positive or B negative if they didn't have the D antigen. So we identify the presence of the antigen. That's what we're going to be doing in our simulated blood types lab, is identifying the presence of the antigen. So what about the antibodies? Because that becomes important. The reason the blood typing is important is because if someone needs blood transfusion, then we need to make sure we know what antibodies are in their plasma, and we don't provide blood cells which have the antigen present, because those blood cells are going to be clumped together. So it's a little unclear to science, medicine, when and why exactly antibodies against A and B are formed, because infants develop them very shortly after birth, okay? and antibodies, there could be blood transfusion issues or blood matching issues in a baby before they're born as far as A and B, but they're not very common. And that's because of antibodies traveling from the mother to the baby rather than the baby having its own antibodies. So it's thought to be, for A and B, it's thought to be linked to plant molecules known as lectins that are very similar after being digested to the A or the B antigen, not the D. We're just talking about A or B. And therefore, if you have A antigen and you absorb plant lectins that are B, that doesn't match what your cells, your lymphocytes, that we're going to talk about next week, have been trained to recognize as self. And therefore, the immune cells, the lymphocytes, specifically B lymphocytes, will recognize that as foreign and develop clones of memory cells that make antibodies against the B. So anytime you have those antibodies in your body already, and you have cells produced to make more, so if you get a transfusion that has the B antibodies, you're going to clump those together. So for instance, let's just look at a red blood cell, a series of red blood cells that have the A antigen. And this person does not have any A antibodies because their immune system has recognized that A antigen is a self-antigen and therefore hasn't mounted an autoimmune response. And then we introduce blood that has B antibodies. We introduce blood that has B antigens. So these B antigens are foreign to your body, right? And because you have ingested, and that's the current theory, plant lectins that had the B antigen that was foreign to you, you have the antibodies already. So now these new red blood cells have been introduced to the body, and you already had, as if you have been vaccinated against the disease, you already have antibodies against the B antigen. So these are what we call anti-B, or antibodies against the B antigen. And when we introduce blood cells to your blood that have these antibodies, they're going to, the Y part of their molecule is going to bind to one cell or two cells, okay? So in the structure of an antibody that we'll talk about next week, this Y formation, this is the area that recognizes a specific antigen. So the B lymphocyte was triggered when you were very young, and ate the B lectin to make these B antibodies. And so this is the binding region, and it binds to that B antigen. And it clumps these cells. That's not a clot, okay? A clot or coagulation occurs when fibrin threads are formed. And so this process is identified as agglutination. So agglutination is the specific clumping of red blood cells by antibodies. So this is the transfusion response if you get the wrong blood type that matches antibodies in your blood.
Can you repeat that definition one more
Uh-huh.
time?
So agglutination is what I showed right here. It's the clumping of red blood cells by antibodies that have recognized the foreign antigen on the cell surface. So this antigen is foreign to this individual's blood type. Therefore, they have antibodies against it. Okay? Again, the reason why is that at some time when they were very, very young, they ingested some form of molecule that their body recognized as being foreign and developed antibodies that would recognize that molecule anytime the body was exposed to it. They probably didn't eat red blood cells, okay, so it's thought to be some form of a plant lectin that the antibodies recognize the same as the antigen on the red blood cell. So your blood type in this particular situation has the A antigen. Your body has no antibodies against A because your body recognizes yourself as self. But you have lymphocytes that have produced antibodies against B. So anytime you're given blood that has B antigens, your antibodies that are already there are going to start pumping them, and that's agglutination. response to the clumping, the red blood cells break apart, they lyse, and it can cause anything from fever to death, okay, as a response depending on how large, how many cells have been caused to rupture. So what we're gonna look at then is based on the chart that you have in your lecture notes. So this image right here is just showing the agglutination, okay, and the subsequent rupturing. So what form of anemia would this be? If you lose a lot of red blood cells due to the rupture caused by agglutination? Yes, hemolytic anemia. And I didn't save this correction from last night, so let me just put it down. I think that's it. All right, so we're gonna go through this table, which is very tiny for you to see, and we're gonna diagram it on, or I'm gonna diagram it while you follow along on the board over here. And we'll do the easy part first, okay? The easy part is identify from the antigens that are present what the blood type is and what antibodies. Agglutinin is another word for antibody. Agglutinin, okay? It's just another word for antibody. But the key point to this exercise are the last two columns, and that's who can you donate your blood to, and what blood type can you receive, okay? Obviously, we're selfish, we're interested in saving our own lives, so we're most interested in what blood type can we receive, but if you're out there donating your blood, it's important to the other person. All right, so, blood type is going to be identical to the antigens that are on the red blood cell. That's how we determine blood type, all right? So then we'll put antigens and antibodies. Actually, I'm going to move this over because I want the room is on the far column, so let me make this a little bit closer together. So we're going to do the D antigen last, all right? It's also known as the RH factor, or plus or negative, as I indicated earlier. So, I'll switch colors and match what's on the board. So if we have blood type A, and I'm going to cut back, as I said, and do positive and negative, okay? So if I have blood type A, my antigen is A. If I have the antigen for A, my body is going to have pre-made antibodies against B, okay? All right, that sets the scene. Now we just follow the scenario. If I'm going to leave a gap here for the positive and negative ones. If I have blood type B on my cells, on my red blood cells, I will have the antigen B, and I will make antibodies. I will have antibodies present that I've made since I was very little against A. Okay. If my blood type, I'll slow down a bit here. If my blood type is AB, and I have antigen A, and I have blood type A and blood type B, so I have both of those antigens. All right, since I have both the antigens, because I would be making antibodies against my own antigens, all right? So blood type AB is not going to have any antibodies. Clear so far? Okay. So you're not going to make antibodies against yourself. You cause your own blood to clump and rupture. You have antibodies against whatever antigen you don't carry on your own blood cells.
So O can't be an antibody.
O has, well, let's work at it. So blood type O, what antigens are present in blood type O? Just as O itself, without thinking about positive or negative, there are no antigens present.
No.
Otherwise, we would say A or B. So we have O, which means none. So what antibodies are we going to have? A and B. Okay, because we always have antibodies for the AB component against the antigen that's not present. Since both antigens are not present, we have antibodies against A, and we have antibodies against B. Okay? Everybody with me on that? All right. So now the next line of questioning is who can we donate to? So I usually use the scenario of sharks in a swimming pool. Okay? Your blood is the pool, and your antibodies, sorry, the other way around. The recipient's blood is the pool, and their antibodies are the sharks, if we're donating to. So if I have blood that has B antibodies, B sharks in it, I don't want to give them any blood that has the B antigen. Okay? So if I'm B, and I'm jumping into a pool of sharks that only eat B, I'm in big trouble. Okay? So we want to look at the antibodies that are in the blood to whom we're donating. And if there's antibodies against us, we can't donate to that blood type. Okay? So if my blood type is A, can I donate to A? Yeah, so we're going to match. That's all we're concerned about. We're going to match completely. There's no problem with A, all right? If my blood type is B, can I donate to A? No, because there are sharks in their blood that would clump my cells together after I was injected, my blood was injected into them. Does that make sense?
Yeah.
Okay? So every single one of my red blood cells would eventually become clumped together or agglutinated by their antibodies. Okay? Can I donate my A blood to AB?
Yes.
Are there any sharks at all? No. So they have no sharks at all. So I can donate my blood A to AB. Okay? Now, I think I know where you're coming from. I have antibodies against their B, but my antibodies going into their pool are going to get diluted out. It's not a situation that's going to, it's not the best scenario, but it's not going to make the other person sick. Okay? Because my antibodies will be diluted in their blood. I'm adding a few, small amount to a huge, larger pool. All right? So I can donate to A and to AB. All right? You're going to see that as a thing. AB can donate to everybody. Not AB can donate to everybody. All of these blood types can donate to AB. I said it backwards. All right? So now we have blood type B can donate to itself. Can it donate to blood type A? No, because there are antibodies against B in blood type A. You can always donate to yourself, but you can't donate to someone else that has antibodies against you. If they don't have your antigen, they have antibodies against you. Okay? So I can donate to B, and there are no antibodies against B in type AB. So I can donate to AB. I can definitely donate to AB here, right? Because I'm donating to myself. I can donate to A and to B. Can I donate to A if I have AB blood? I have A and B antigens. If I donate to A, do they have antibodies against me?
Yes.
Yes, they do. If I antibodies against me? Yes. So AB cannot donate to A, cannot donate to B. Can I donate to O? Worst case, they have antibodies against both of my antigens. Okay? So I cannot donate to O. So just to AB. All right. O. Can O donate to A? Yep, because O has no antigens. Therefore, no blood type has antibodies against it, right? Now, yes, it has antibodies against A, but remember I said, although not desirable, it's not going to cause this person to get sick. They're going to be diluted out. So O can donate to A, O can donate to B, and O can donate to AB, and O can donate to itself. Okay? Does that make sense? So you have to ask the same question each time. In the blood that I am donating to, are there antibodies in that blood that are going to cause my cells to clump together? And since O has no antigens, it doesn't matter if there's antibodies present or not. So you can always donate to yourself. And there's no A antigen on O. There's no B antigen on O. So I can donate to A, B, or AB. Okay? All right, next question then is, what blood type can I receive? So if my blood type is type A, what blood type can I get? So it's a similar question, just in reverse. So I can always receive from myself, okay? Then it's, what antibodies do I have? If I'm A. B. So I can't receive anything that has antigen B on it, because I will clump all of those red blood cells coming to me. So now I'm not concerned about the other person's antibodies, I'm concerned about my antibodies. I'm the receiving pool, okay? So if I have blood type A, I can receive from myself. I have B antibodies. So may I receive from blood type B? No. May I receive from blood type AB? Nope, because I'll agglutinate the B. Can I receive from blood type O? Yes. Even though O has antibodies against me, again, they will be diluted out. Okay? So I can receive from O. All right? So can I receive blood, if I'm B, can I receive blood from B? Yeah, I can always donate to or receive from my same blood type. However, I have a whole pool of antibodies against A. So I can't receive blood type A because I have antibodies against that antigen. And again, I can't receive from AB because I have antibodies against the A part of AB. But since O has no antigens, I can receive from blood type O. Okay? All right, blood type AB. I can receive from myself. Okay? Do I have any antibodies? No. So, I can receive from A, because I have no antibodies against A. I can receive from B, because I have no antibodies against B. And I can receive from O, because there's nothing to have antibodies against if you're O. Okay? Now, any questions on that? Okay, you just have to remember which way you're looking at this side of the coin as far as where the antibodies are. All right, if I have blood type O, what antibodies do I have? Both A and B. So can I receive A? B? AB? Nope. So all I can receive is myself, and that's blood type O. Okay? So now we add the third antigen, And this one's a little bit different. It also appears in the blood when your lymphocytes have been exposed to the D antigen or the rhesus antigen. But it doesn't seem to have any plant lectins or anything else out there that contain the D antigen or anything similar, except someone's blood that has the D antigen on it. So there has to be blood exposure for individuals to develop the D antibody, okay? Some people have the D antigen, most people have the D antigen, but not everybody. So we're gonna use the D antigen with red plus. Last night we switched it, it was green, but anyways. So if you have the D antigen, your blood type is A positive. If you have the B antigen, well, let me just go through and write all the other ones on there. We'll just do it by. All right, so if you don't have the D antigen, I did it backwards here. Your blood type is A positive, you have the D antigen. If you don't have the D antigen, your blood type is A negative, okay? If you have the D antigen, your blood type is B positive. And if you don't have the D antigen, your blood type is B negative. A, B, and D antigens means your blood type is AB positive. Don't have the D antigen again, your blood type is negative. If all you have is the D antigen, your blood type is O positive. And if you don't have any antigens at all, your blood type is O negative. Okay? Now, before we look at who you can donate to and receive, we have to understand the antibodies, right? So, if you have the D antigen, will you ever have the D antibody? No, because you would be making antibodies against yourself. If you don't have the D antigen, would you all automatically have the D antibodies? Only if exposed. So if you're male, only if you've had a blood transfusion, okay, of somebody with positive blood and you are negative. If you're already positive and you get a blood transfusion of positive, you're not going to make antibodies against their D antigen because you already have it. If you're negative and you get a, say you're A negative and you get A positive transfusion, you will make the antibodies. Not right away, it takes a couple weeks. All right, the process starts after 3 days. It takes a couple weeks to be able to measure how many antibodies are actually in the blood. However, if someone does not have antibodies, all they have is the B antigen, they have A antigen and all they have are B antibodies and they don't have any D antibodies, do I know whether they're positive or negative by just looking at the presence or absence of the D antibody? No, because they could be B negative, they could be A negative, just never having been exposed to the D antigen. So the absence of the D antibody doesn't tell me whether they're positive or negative, okay? The presence of the D antibody tells me that they're negative. So, and the antibody is going to appear only if exposed. So if I tell you somebody has A and B antibodies, you would know that their blood type is O. Do you know if it's positive or negative yet? No. But if I say they have A and B and D antibodies, the presence of the D antibodies tells you their blood is negative and they have been exposed to the D antigen. All right? So if someone has blood type B positive, then they would have antibodies against A and they would not have any antibodies against B. Their blood type is B negative, they would have antibodies against B and anti-D only if exposed. antibodies. And if they're AB negative, they're still not gonna have A or B. And again, only the D antibody if they're exposed.
I have a question.
Yeah.
In the beginning, you said we receive the D antigen only by exposure as well?
And antibody. The antigens are
So-
determined genetically.
Okay, that's
So the A and the B antibodies, so if you have A antigen, you would develop the B antibodies. The theory is based on food that you've eaten when you were very, very young. Or then, same with B. The D antigen is genetically determined. If you're negative, you would never make the antibody if you have the antigen. You would be making antibodies against yourself. You would only make the D antibody if you were exposed to blood that had the D antigen.
Okay, so this is why you have to do blood typing often, because it can change.
Yeah.
Right?
If you're negative.
Yeah, okay.
The blood type won't change, but the presence of the antibodies
This
will.
will, okay.
Yeah.
I always wondered why they did it more than once to one person. I'm like, hey, that makes sense.
All right, so if your blood type is O positive, you would have antibodies against A and antibodies against B, but nothing against D. If you're O negative, again, you would have antibodies against A, antibodies against B, and again, only antibodies against D if exposed. Okay?
If you haven't been exposed yet, can you test out to be negative?
Okay, say that again.
If you have negative Rh factor,
Uh-huh.
I haven't been exposed to the antigen yet. When the blood test is already show I'm still negative. I'm negative with Rh factor.
I still don't understand the question.
Can
So if you're negative,
can you test somebody before exposed?
yeah, you can test for antibody titers. Just like if they can test to see if your smallpox vaccination or your chickenpox vaccination is still working, they'll test to see what the levels of antibodies are in your blood.
You're still negative, but you just don't have the antibody.
I'm not sure what you mean by active.
No, not active. You don't have the antibody yet,
Right,
but you are negative.
you're negative, right. So you're negative, you could say negative your whole life if you were never given, if you're a man and you were never given a transfusion, or if you're a woman and you never have a baby who is positive, or a pregnancy, I should say, okay?
I have a question about
Uh-huh.
the DSR. So if you're exposed to it, you just get the antibody, or do you get the agglutination too from D?
You get what kind?
Like, if you're exposed to D and you're negative, does agglutination happen, or it just?
It depends. So it takes 3 days to become primed to make antibodies. If it's the first exposure, it can take up to 2 weeks to actually make any.
Okay.
Red blood cells have a lifespan of 4 months. So you would probably still, depending whether you got 1 unit or 3 units or whatever, it would depend on how many of those cells you actually had. So you could get a delayed reaction, but it wouldn't be nearly as strong as if those antibodies were already there. Because in that first primary immune response, which you'll see next Thursday, it doesn't get nearly as high as the second one, and it drops off much more quickly.
Like the second exposure was
Second
already
exposure,
happened.
yeah.
Okay.
Yeah. All right, so before we look at donate to and receive from, we need to think about the fact that we don't want to cause someone who is negative to have a primary immune response and develop the antibodies. So we want to keep them virgin in that sense, all right? Because they might need, or they might get a blood type that's positive by mistake, or that might be the only blood type available. Let's say they're AB negative and the only blood type available is AB positive. And to have any chance of living at all, they have to get that. So if they don't have any antibodies yet, we could save their life. They would never be able to get it again, but we just want to avoid that whole situation. So we never want to receive positive blood if we're already negative, and we never want to donate if we're positive to somebody who's negative. All right, we want to keep the absence of antibodies to be a continuous component. So if I am AB positive to can I donate? Well, first of all, just to save time, let me go through and put all these second blood types in here. All right, so if I'm A positive, can I donate to negative? I should, okay? So I can only donate to A positive and to AB positive. All right? A negative, yes.
So you said in a life or death situation, you could do it, but you wanna avoid it. What happens when you do it, you don't?
So that's what we were talking here. So that first time that you would give someone who is negative, positive blood, they would develop what's called a primary immune response. It takes 3 days to start an immune response. After 3 days, they're going to be form antibodies. And then it takes, if it's the first immune response, it's usually a couple weeks before you see a titer that starts to rise. And then it falls pretty quickly. We'll see this next week. Let's say here's the first exposure. And then you'll see a titer, this is 2 weeks. And you'll see a titer, which means the level of antibodies in their blood, that will rise and fall right away. Their second exposure, it's almost immediate, and it stays elevated for several, over a month or so. So you would be at this point right here, if it was their first exposure.
So it's not permanent, it's just.
No, they would be permanent. The second time they get exposed to that blood type, to that positive, they would immediately have a transfusion reaction. Because they would have that high level already in their body.
Oh, so they wouldn't be able to do it for
Right.
Okay.
Yeah. So I'm just saying, we want to keep that possibility there and not trigger an immune response in them if we don't need to.
Okay.
Now, once they have the immune response, they're always going to have antibodies against the D antigen. All right, so if they are this here. If they are A negative, that negative means they don't have the D antigen. So is it going to be harmful to give somebody that has a D antigen blood that doesn't? No, it's a cipher, it's an absence, okay? So we can give negative to both positive and negative blood. So since we already decided that they could donate to A and they could donate to AB, then negative can donate to positive and negative can donate to negative, okay? So if you're positive, you only want to donate to positive. If you're negative, you can donate to either. So B positive can donate to B positive and to AB positive. B negative to either positive or negative. Same thing, if you're AB positive, you can only donate to AB positive. If you're AB negative, either or. If you're O negative, everybody wants you to come to donate blood to their blood bank. So you can donate to O positive, O negative, A positive, A negative, B positive, B negative, and AB positive and AB negative. All right? So O negative is identified as the universal donor. So my husband is O negative. They're always asking him to come down and donate blood. All right. Any questions on that? Again, please don't memorize this table. I'm going to gonna ask questions on it, definitely, but if you know the principles behind, then you can always figure it out, okay? All right, if you are A positive, can you receive positive blood? Yes. Can you receive negative blood? Yes. So, if you are A negative, you only want to receive negative, okay? So it's the flip side of this coin here. If you are B positive, you can receive positive or negative for B or O blood. If you are B negative, again, only the negative version, okay? If you are AB positive, you can receive AB positive or negative, A positive or negative, B positive or negative, and O positive or negative. So this is the universal recipient, okay? Safest blood type to have as far as what you can get. It's not as common as O positive, but you can receive any blood type, positive or negative. Now, although you can receive any blood type, you can only give to yourself, okay? Now, O positive can receive O positive or O negative, and our altruistic O negative that could donate to everybody can only get O negative. So altruistic type O negative has to find itself its own blood type as a transfusion source. Okay? Does anybody know what type of cross-match means? We're going to be doing the typing part in lab today, all right? So this doesn't have the D antigen on it, but antibodies against A or B were added to 2 samples of the same blood. So this blood type here, both of them are the same person's blood. Antibodies against the A antigen were added, and then a different solution with antibodies against the B antigen. We're not worried about the antibodies that are in the blood. We're using antibodies to find the antigens that are already there. We're totally ignoring the antibodies that are already in the blood. What we're trying to do is find out what antigens are in there. Kind of like going fishing with a fishing pole and a magnet in a wash tub that has little fish that have magnets attached to them. You know, if you've got a 5-year-old birthday party or 3-year-old birthday party, and they throw in the magnet and it binds to a fish magnet and they pull it out. So we're throwing in antibodies to see if they find an antigen and cause the blood cell to clump. So if the blood cell clumps, the antibody against A found A antigens. If the blood cell does not clump, the antibodies we added, external antibodies, not antibodies that are already in the blood, the antibodies that we added did not find any A antigens. So here they did, here they did, here they did, here they didn't. So on this side, B antibodies were added. Outside external source, found the B antigen, found the B antigen, did not find the B antigen. Okay? So here, both A and B antigens were found, so the blood type is AB. Here, neither antigens were found, all right, and the blood type is O. Agglutinogen is not the same as agglutinin. Agglutinogen is the big long fancy word for antigen. So agglutinin is antibody, agglutinogen is antigen. Okay, so don't get mixed up by that. So I'm going to turn the screen blank. So if I give you a situation where the blood that we're looking at has antigens A and D, what would the blood type be? Pardon? A positive.
negative if it's been exposed?
Got the antigen,
Oh, okay.
okay? So it would be A positive, right? If there were no antigens, what would the blood type be? O negative. Okay, so we're just looking at antigens right now, all right? If they had A and B antigens, maybe negative. Okay? So we're doing our lab blood typing today. We're looking for the antigens, and from the antigens that are present, we're getting our blood type. Okay? So if I were to diagram what this would look like, we're using plates that look like this, not this big, and there's oval depressions. And if we put the same, we're gonna take one simulated person's blood and put a couple drops in each one. And then add, a lot of people yesterday didn't have very much in there, so it was hard to break the surface tension and get the blood to roll back and forth to see if it was agglutinated or not. So put a couple of drops and then 3 to 4 of the typing solution. So you have enough fluid in there that you can roll it back and forth, all right? So we add antibodies. We have a dropper bottle, and we add antibodies against A here. And we add antibodies against B. And in the upper corner, we add antibodies against the Rh factor, which is the same as B. And then you'll get toothpicks to stir the solution, all right? Don't use the same toothpick point in all 3 of them, or even in another tray, because we don't want to cross-contaminate. So you want to either use the opposite end or just put it aside and get another one. So if we have this blood type, are we going to find A antigens? If we have A antibodies? Yeah, we're adding A antibodies, and they're going to find the A antigen. Okay, so we said here, we have it. So as a result, this blood type is going to agglutinate. And we have the D antigen. So that also will cause agglutination, because the antibodies against the D antigen are going to find the D antigen and cause agglutination. Okay? Then we add antibodies against B for this depression of the same blood. Are there any D antigens there? No, so it will not agglutinate. So if we see this result, we can say A is present, D is present, our blood type is A positive.
So it's not agglutinating against, it's agglutinating for.
Right, we're looking for the antigen,
For,
we're
okay.
fishing for the antigen. So we're not concerned about the antibodies in this blood at all.
Okay.
We're using external antibodies for the antigen, okay? All right, so for blood type that's O negative, do we find A? If they're O negative, do we have the A antigen? Nope. And we don't have the B antigen, because we're negative, and we don't have the B antigen. So the blood, none of the depressions are gonna show any agglutination. So you can say, I don't have A, I don't have B, I don't have B, therefore blood type is O negative, okay? If blood type was O positive, then you would have no A or B, and you would see agglutination only for the D antigen because your blood type was positive, okay? So our last situation here, blood type was AB negative. So we have A, we have B, we have no B, okay? So that's how we're going to translate or read the typing tray, okay? Now, I'm gonna flip the story on you again, okay? May I erase that part of the board? determine what the blood type is based on the presence or absence of antibodies, okay? So if we have antibodies against B, and we have antibodies against D, what would the blood type be? We would have A, and we would have no D, but the person would have been exposed to it, all right? So blood type is A negative. Okay, so the presence of anti-B means we don't have B, we have A. The presence of anti-D means we don't have D, which is why we put negative, okay? All right, let's say you have no antibodies. Do we have A? Do we have B? So blood type AB. Do we have D? Maybe, maybe not. If we did have D, we would have no antibodies. If we didn't have D and there was no exposure, we wouldn't have antibodies either. So all we can put here is a question mark. Okay? I don't know. All right? All right, I have antibodies against A. Do I have the A antigen? No. Antibodies against B. Do I have the B antigen? No. No antibodies against D. So again, I'm back to, I don't know. Okay? So I could be positive and never get antibodies, or I could be negative and never have had the stimulus to make antibodies. Okay? So again, practice this to understand the principle, but please don't write the table 5,000 times to memorize it. That's using up brain space you can devote to something else. So understand the principle, but not memorize that table.
Is this just looking at blood type based on antibodies?
Right. Yeah. So these are antibodies that are in the blood. We're not doing that lab thing.
Yeah.
So you kind of have to figure out. That's what you kind of have to think through. Where am I? You know, what part of the kind of like identify a muscle? Am I looking at an arm or a leg type of thing? Okay. All right. So then the last concept before we take a break and start lab after your break is a condition I've alluded to. That refers to fetal, not fatal, okay? We're talking about babies, although it often is fatal. So erythroblastosis fetalis is a condition that occurs when the mother's blood type is negative and the baby's blood type is positive, okay? So before we look at those specific situations, let's look at a genetics chart here. And we'll put the mother up here and the father here, okay? If the mother is, write it up there. If the mother is homozygous, positive, all right, she would be dominant. R is positive is dominant. If the father, homo just means both genes, one from each parent, is dominant. If the father is homozygous positive, meaning they have the D antigen, they have genes from both parents, then all of their children will have a 100% chance. This is just a probability for each child. Each child has a 100% chance of being positive. They're going to have the D antigen, all right? So this will be no problem. Erythroblastosis fetalis will not occur, okay? Now let's say that the mother is heterozygous, meaning a dominant gene and a recessive gene. And the father is heterozygous. chance that the child will be positive. We're not saying 3 out of 4 children, we're saying each time a child, the sperm enters the egg, there's a 75% chance that there will be a dominant gene present and the D antigen will be formed. Each child has a 25% chance of being negative. The mother is positive, right? Doesn't matter what the baby is, the mother's positive, no chance of the baby developing urethral blastosis fetalis. Okay? The mother has to be negative. Does that make sense? Because it's mother producing the antibodies. If she's positive, she's not going to be making antibodies. All right. Now the mother is homozygous negative. That's a recessive gene. And the father is homozygous positive. Even though the mother is negative, all of her children, if she's married, not she's married or not, if the father of the children should be having an affair for that matter, the father of the child is homozygous, she'll never have a positive child. So even though she's negative, no matter how many fathers of her children she has, if they're always positive, homozygous, she doesn't have any chance of having, let me rephrase that. I just said it completely wrong. Comes from being in work all day. So she's negative, and her father is homozygous positive, all of her children. There's a 100% chance, backwards what I say. All of her children, each child, every time, will be positive. They will be heterozygous, but they will still have the D antigen. All right, so in this case, here's the mom. Urethral blastosis. I put the head all upside down because babies are born head first, but anyway. The first baby will be okay because the exposure occurs at the separation of the placenta and the decidua basalis and the tearing of the maternal blood vessels. And some of the baby's blood cells will get into the mother's bloodstream. Remember I said it takes 3 days to complete that exposure in the mom's response, and any pregnancy afterwards, she will have those antibodies already in her body. And her second baby, second positive baby, if her second baby is negative, there won't be a problem because there won't be any antigens for her antibodies to bind to. Her second baby that has the D antigen, however, the mother's antibodies can cross the placenta, and the baby's red blood cells will be agglutinated. This doesn't always mean death, but these babies, if they don't die in utero, and they often do, we're first treated by giving them transfusions after birth. All right? So if the mother is A negative, and the baby is A positive, because the dad was positive, the baby got the dad's dominant gene here. If the baby is A positive and the mom is A negative, this is her second pregnancy, all right? The baby was born still alive, but very sick. A lot of its red blood cells had already been agglutinated. And we give it a transfusion. Do we want to give it an A negative transfusion or an A positive transfusion? A positive. Remember, A negative has no D antigen. If we give it an A positive transfusion, we're adding more D antigen, and it still has its mom's antibodies in its bloodstream. And it's going to keep making more of its own A antigen anyway. So we're going to give it A negative. Therefore, any antibodies still remaining from the mom will have nothing to attack. Okay? Because A negative just means it's nothing, right? The D antigen is absent, so we're not introducing anything for it. We're just leaving out the D antigen that the mother's D antibodies might attack. Okay. Last situation. Mother is still negative, and now the father is heterozygous. baby has a 50% chance of being negative, which in case everything's fine. Because it's negative, the mom's negative, all right? But the baby has a 50% chance of being positive. So in this case, any baby that this mom had with a homozygous D antigen dad is going to be positive. Here, only a 50% chance, okay? All 4 of her kids may be negative because we're talking about the probability of each pregnancy. We're not multiplying. Here, each baby has a 50% chance. However, if the baby loses the lottery, and again, is the second positive baby this mom has, it will have antibodies crossing the placenta and causing agglutination. Well, we obviously couldn't save the babies that died in utero by doing a transfusion after birth. So, medical science started doing blood transfusions before birth, in utero, into the baby's abdominal cavity. And that still didn't save all the children, all the infants. And so, back in the late 60s, early 70s, as soon as the baby was born, and his blood tested positive for the D antigen, and it was a positive baby, the mother was given a RhoGAM shot within 72 hours.
When they also give it to you, while pregnant, they don't even take.
They
the
do now.
blood anymore, yeah.
All right, so the RhoGAM is the antibodies. Or we've been writing it anti-D. Remember I said it goes to the mother? So if this is her first baby, and the baby's blood cells that have D antigen on them enter her bloodstream, remember I said it takes 72 hours for her to finish that primary immune response. So that's why this has to be given within 72 hours. And what it does is it clumps and agglutinates all of her baby's blood cells that are in her blood before she mounts that primary immune response. So the next time she has a positive baby, it's as if it was her very first, okay? And you're right, now they give the RhoGAM shot during the pregnancy, I think twice,
During the pregnancy and 7
and then
days
afterwards.
after.
Okay, once during pregnancy?
I think it was.
So it doesn't matter, they don't even try to type the baby, it's
Yeah.
just in case, we'll go ahead and do that. And so this is attacking the baby's blood cells, causing them to be destroyed before the mother makes her own antibodies. So she doesn't have any plasma cells that have already cloned and made antibodies, okay? So it's a preventive method for the next pregnancy. Now, there's been several situations, so this is not exactly made up, but I'm not thinking of a specific one offhand. A mom has a baby. The baby is positive, okay? Baby, well, let me rephrase it again. The baby's negative, so obviously the mom doesn't have any problem. The mom has another baby. That baby is positive and is born with erythroblastosis fetalis. Now her first baby is negative. He's 2 now. This other baby last night, they were both girls. This time they're boys. So the first baby is negative. Second baby to be born is positive and already has erythroblastosis fetalis. Fortunately, didn't die in utero. Mother never had any transfusions. She didn't need any transfusions with the birth of her first baby. She hasn't been exposed to any blood that has a D antigen. So how does she develop antibodies? Yeah. In this particular case that I read about, the baby was the first baby, a little boy. Obviously, the adult was a baby. The first baby was one of twins, actually. And fraternal twins, because he was negative and the other baby was positive. And the baby, the twin, died so young that the OBGYN didn't even pick it up. So it could have been within the first 3 or 4 weeks. They already have blood cells, and the baby died, and the baby's fetal tissue degenerated, broken down, absorbed back into the mother with cells that had the D antigen. And she was never aware of miscarriage. You might be aware of. But it was still a miscarriage. I mean, the baby died, and she became exposed to the D antigens on the absorbed tissue as the baby tissue was broken down in utero. Okay, so there can be cases where, yeah, you are pregnant and not aware, unless you're testing yourself every 3 days. Yeah.
So what if it was like your first pregnancy, you had a miscarriage, and you obviously didn't get the would they give you the rhodium shot, or would you obviously not get it?
Well, you would have gotten it. It depends. You would get it later in pregnancy, and so it still could save that second baby.
Oh, okay.
Well, no, it would be too late. Yeah, it would be too late because the mother's immune response is already, she already has the antibody.
So what do they do?
But I would assume, I haven't worked in the medical area for a while, I would assume that antibody titers, especially if she had a series of difficulty getting pregnant,
Yeah.
because there's several different causes. There are women who have antibodies against the dad's sperm.
Just for the ask how many miscarriages you've had, like,
Yeah.
pregnant.
So if it's been a lot, then they might start looking
I had it.
for the antibody levels to say, oh, you know.
Yeah.
And then, unless you have a negative, you might do in vitro fertilization or intracytoplasmic sperm injection to avoid the antigen. Okay? All right. So, yeah.
So does this hurt the mother?
No, because it's the baby cells in her that are being agglutinated, and there aren't enough to cause a transfusion reaction. So she's not really getting, I mean, if babies are born and they're held above the mother's heart, then you can get a lot of blood transfused back to the mother. But typically back to the placenta, I should say, not to the mother. But if in that separation, there's not enough of those fetal cells entering the mother to, if they're agglutinated, to result in a transfusion reaction. You only need, a few. You only need one plasma cell, one B lymphocyte to get a reaction, to get them cloned, and have numerous ones in the body. Okay. All right. So I have given this.
So like when the agglutination happened, like when exactly is that? Like,
So
how many
for
weeks?
this or for the
Or
baby?
like if there's a second copy
Okay.
of that?
So the mother's antibodies are already present in her blood. Okay. And I'm not saying it's impossible for the baby cells to leak into the mother because there can be some kind of a primer. But the mother, you don't usually have a lot of antibodies unless there's an exposure event. But the baby's there for 9 months. So during that entire time, antibodies that the mother already has circulating in her system, although they're low, can move across the amniotic membrane and in from the mother's blood into the fetal capillaries. All right, so before I bring in the blood typing and go through, although we already went through the steps, I have a tray of human blood cells here. They are whole, in other words, they haven't been put in plastic in sections, and they have been spread, they have a dye on them, so they look dark blue as far as the stain is concerned. All right, so they're under a cover slip. And when we look at red blood cell, not red blood cells, when we look at try to identify white blood cells, they're pretty small, okay? Not as small as the sperm we were looking at, but not nearly as big as the eggs for the sea urchin. And so, to help us be able to see them larger, we're going to use oil immersion. Anybody here that's used oil immersion already for micro? Yeah. Okay, good. So if you're scattered around, you can help your classmates. And what's the purpose of putting in a small drop of oil? Our lens is going to do the magnification. What we're trying to do is keep the resolution good. For instance, if I take a image, a black and white image, and I put it on a copier and I magnify it 200 times, and then I take that picture and magnify it 200 times, I'm going to keep getting it bigger and bigger and bigger infinity, but it's going to start having blurred edges. I'm gonna lose the resolution, I'm gonna lose the ability to distinguish a sharp edge. And that becomes a factor when we magnify 100 times. We get blurred edges because light waves are not always focused on the same spot. When light passes through glass, depending on the shape of the glass, it's gonna bend one way or the other. And it's gonna bend again when it gets into the air, because they have different refractile indices. And so when we bring in our oil immersion lens, it's going to come in and actually enter into the oil droplet. So when the light comes through the glass slide, through the tissue, through the cover slip, through the oil, into the objective, it never goes through air again. So it maintains, it doesn't bend back and forth, and we maintain the same first bend so that we don't have overlapping images occurring on our lenses. And we maintain a higher degree of resolution. We can see sharper lines. So we can magnify more without getting fuzzy edges. Now, because it's oil immersion and we're doing this higher magnification, we also have a very long, narrow tube for the light to go through. You might see that you have to increase your light intensity, okay, or open up the iris, because this is cutting down the amount of light. Because we want all these light waves to be parallel, again, so we don't have fuzzy edges. So we're trying to narrow the amounts, just like rolling up a piece of paper and looking through a telescope. Edge that decreases light waves that are slightly at an angle. Most of them are going to be in parallel, and that also helps to make the image sharper, but it means it's darker because we don't have as much light coming through. Okay, so the 100x, or the oil immersion, it might be 93x, is sealed against the oil. 40x is not. All right. So when you set this up, you want to focus without the oil with the 40x. You'll be able to see the cells and focus. They'll just be really small. If you want to look at them that way, you're welcome to. They're just small. And then turn the 40x slightly away so that the oil immersion is not all the way in place. You have an open space there. Add your oil and then double check twice to make sure you're bringing the oil immersion in and not the 40 back into place. Okay? So once you put the 40 in there, the scope's going to be ruined until the summer when they get all cleaned again. So double check before you put the 100x into the oil, and then you can look in there and scan them. So what you want to do to make sure that you find good areas of cells is, if they look all ruptured and clumpy, just get a different slide, okay? Stay away from the edges, because they'll tend to be piled on top of each other there, the red blood cells will. And then you can go up and down this way, or you can go crisscross, but you want to kind of scan through in a pattern. You're not looking at the same area. Now, we're not going to do counting, all right? Sometimes I have students count up to 50 to see what proportions they have, but I want you to just get some practice at finding them in their natural living situation until they leave the blood at least. And so I'll put the slides over here while you're getting set up for the typing. So let me go grab the typing. I'm going to tell you how to do it, and then you can take a break or work on that as you will. And I'd like to start lecture at about 8:30. Okay? I'm sorry, it says 7:30, not 8:30 now? Yeah. Okay. We started later than last night.